Hello and welcome to the second season of Digital Science for You. Cyber School Technology Solutions is an organization that's revolutionizing education. They've got a software, digital science, and virtual lab. The software has 6,326 videos and animation. It covers around 120 topics of the curriculum and has 157 experiments in their virtual lab component. All this comes with a package of quizzes and tests, if you like. And as a student, I advise you to get yourself this copy. To get it, you can head over to Uma Showgrounds Logogo and look for Cyber School Technology Solutions, or head over to social media and look for CSTSUG. So sit back as a student and let's do some learning. One of the reasons we study biology is that ultimately it's life. You study about plants, you study about human beings, you study about other animals. So biology is important to your life, like it or not. But why do we say that it's one of the hardest subjects to understand? Maybe because it's hard for teachers to draw the drawings of all these organisms on the blackboards. Well, guess what? Coming up is teacher Joan Namubiru. She's using digital science and virtual lab to help you, a student, understand biology better and easier. And while you do that, you're going to have fun. So sit back and let's do some learning.
Dear students, you're welcome to yet another biology lesson by teacher Joanita Nambiru. We are going to be studying biology and we are going to be covering a topic of senior 2 known as nutrition, and specifically we are going to be talking about enzymes. We are going to be using digital lab and we are going to digital science, and we are going to be using virtual lab to help us understand these concepts. So by the end of this lesson, you should be able to define an enzyme. You should be able to describe the properties, some of the properties of an enzyme, explain the factors that affect enzyme activity, and we shall go to the virtual lab and carry out one experiment. So let's get started. You are very, very welcome. Okay, let us go to senior 2 right there. We're going to use digital science at first. It's biology. And our topic is nutrition in plants and animals. We're going to go to nutrient compounds. We're going to show, and then we are going to go to 2 enzymes and their properties right there. A lot of things happen in our bodies. A lot of processes happen in our bodies. You have already learned about digestion. Probably you've done digestion, but you know that digestion is actually carried out by chemicals known as enzymes. We have a lot of processes in the body, including respiration. Plants carry out photosynthesis. All those processes occur by the help of enzymes. In your senior 1, for example, in chemistry, you have been doing preparation of maybe gases with the use of catalysts. Now, God was very bright in our bodies. He put there catalysts, and these catalysts are known as enzymes. What do these catalysts do? They basically increase the rate of a chemical reaction within our bodies. But these catalysts are actually proteins in nature. Let us go on and we begin. How do these enzymes work? How do these enzymes work to make sure that they speed up the rate of a chemical reaction? Those are the enzymes. They are catalysts, biological catalysts. Number 1, they are proteins in nature, and they speed up the rate of a chemical reactions. Now, catalysts work on a substance that we call a substrate. Enzymes work on a substrate, and then they combine with a substrate and they form products. Let us look at this and we see what is going to happen. Let's just go back right from the start. In red there, we have our enzyme. In red, we have our enzyme. As you can see, enzyme. The enzyme has got a specific surface, a specific area where it binds with the substrate. I told you the substance on which the enzyme acts is called the substrate. Now, that substrate must be complementary to the enzyme. It must fit to the active site of the enzymes. If it does not fit, if it is not complementary, then the reaction will not take place. So let us continue and see right there. Let's move on and see. All those are substrates in blue and in yellow. They are all substrates. Our enzyme is red. Now, which substrate will be catalyzed, will be broken down? Check that yellow one. That is the active site. The particular site where a substrate binds with the enzyme is called an active site. And it must fit exactly. Check that. Let's see that substrate. Does it fit? No, it does not fit. Therefore, check that. That one fits exactly. Let's first stop there. That one fits exactly. And an enzyme works exactly like the key and the lock. When you know that every door has its own key that opens it. So we have a lock here, and the key should fit exactly into the lock to unlock that key. It may fit, but if it doesn't fit in the active site, it will not open the door. So the enzyme is like a lock, and the substrate comes to fit in like the key. And then when it fits, then the enzyme can be able to lower what you call the activation energy of that reaction, and then we shall have the substrate forming products. Very, very important. I want you to note what happens to the enzyme. Let's go check that. Do you see that our substrate is breaking into two? Now we have two products, simpler products. What has happened to our enzyme? It has remained unchanged. Do you see that? We have the products there. They were at one time it was one complete substrate, but it needs to be broken down into simpler substances, and those are the two products. And this is our enzyme. Our enzyme has remained unchanged, and we have described the lock and key hypothesis, or lock and key mechanism, that describes or explains how enzymes work. An enzyme is a biological catalyst, protein in nature, that speeds up the rate of a chemical reaction within the body. Very important to know that it remains unchanged at the end of the reaction. So when it forms a product, then it can as well go back to pick on new substrates and bind with them to form more products. Let us move on to the next slide. As you can see, they function as catalysts. They speed up biochemical reactions without undergoing any change. Very, very important. We move on. What are some of the factors that determine the rate of a chemical reaction or an enzyme reaction? There we have, let's look at the first factor, temperature, and then the next factor, pH. For today, we shall be more specific to those. Before we move on, let's move on. Let's look at temperature at first. How does temperature affect the rate of an enzyme-controlled reaction? At first, you see that temperature is at 0 degrees, very, very low. What happens? There is completely no enzyme reaction, and that is the description. Now, in biology, we are interested, what is the explanation for that? At that point, we say all enzymes are inactive. You can say, they are not active. Don't use the word asleep, but if you're inactive and you're sleeping, then you're inactive. So at a very, very low temperature, these enzymes are all inactive. They can do nothing. So as temperature goes on increasing, then the rate of reaction continues to increase. As we have further increase in temperature, we can see that the rate of reaction also increases. Why does it increase? Because now increase in temperature sort of increases the kinetic energy of these enzymes and then the substrate. So the higher the temperature, the higher the kinetic energy, then the higher the chances of the enzyme and the substrate binding together along the active site to form the products. So, and then at the point there, let's go to the second one, that one there, that's still enzyme. Effect of temperature on enzyme activity. As temperature increases up to the optimum temperature, exactly that point down, let me bring it down. Yes, that temperature there is what we call optimum temperature. Let's go to the y-axis and see the rate of reaction at that optimum temperature. It is highest there. I think you can see that. it is highest there. That's what we call optimum temperature. So what is optimum temperature? Optimum temperature is the temperature when the enzymes are all activated and they have the highest rate of reaction, the best temperature at which an enzyme can work. And usually for our bodies, that optimum temperature is about 37 degrees. That is the normal body temperature where all activities can go on very, very well. Now, what happens to the enzyme above, optimum temperature? We have said where enzymes work best, and all enzymes have been activated or are activated at optimum temperature. Now, what happens beyond optimum temperature? When temperature increases right there above optimum, you can see that there is a rapid increase in the rate of reaction, to 0. Why? Because now increase in temperature is going to denature the enzymes. What do you mean by denaturing them? It's going to destroy them. Just imagine you have an egg, because an egg is a protein just like enzyme. What happens when you put an egg in water? It solidifies. When you cool it back, you cannot get the shape back. So when you increase the temperature, you're destroying the bonds that are holding the enzymes. So you are destroying it, you are denaturing it. So we say low temperatures inactivate enzymes, high temperatures denature enzymes, and that's why the rate of reaction reduces. Optimum temperature activates all the enzymes, and we have the highest rate of enzyme activity. And that is it for temperature. We can actually do experiments on these, but let's first go on to pH. Still in nutrition, we study about the digestive system. As we can see there, there is a stomach. We know that pepsin is right there. Pepsin is an enzyme produced within the stomach. And when we talk about pH, we talk about, let's first look at the pH meter here down. The pH meter begins from 1, 2, 3, all the way to 14. When we begin from values 1 to below 7, those values are acidic. So when we have a solution with the pH 1 or 2 or even 6.9, all those values are acidic values. If it is at 7 exactly, that is neutral. For example, pure water is neutral, neither acidic nor alkaline. Then when we go above 7 all the way to 14, then the pH becomes alkaline. The particular enzymes work best at specific pH. Particular enzymes work best at optimum pH. So an enzyme may work at a pH which is acidic. Another one may work at a pH which is neutral. Another one alkaline. But if that pH is not optimum for that particular enzyme, then what is going to happen? That enzyme is going to be denatured. And the moment the enzyme is denatured, it can never work. So let us look at the stomach. Like pepsin is an enzyme secreted within gastric juice, and therefore that enzyme works at a low pH which is acidic. Right about this time, let's take a break, and when we come back, we shall finish that factor and we shall go to virtual lab and we shall wind up our lesson.